I'd like to start by acknowledging the traditional owner of the land on which we meet and custodian of the land on which we meet today, and pay my respects to the elders and elders of other communities. So it's a real pleasure to introduce Tracy Leong, who's going to give a PhD completion seminar today. Tracy is a respiratory physician. She's done most of her specialty training here in Melbourne after doing a Bachelor of Medicine in Adelaide. And during her training, she really got some interest into research, and more specifically in how we can use this novel technique of endobronchial ultrasound guided biopsies to really address some questions to better understand advanced lung cancer. And so she's taken really multiple approaches to evaluate how we can use those biopsies, and she's going to talk about 3 of her projects to you today. Tracy has had a bit of an unusual PhD. She started at Monash University with Neil Watkins, and then when Neil moved his lab to the Garvan Institute in Sydney, she transferred to Melbourne Uni. And then she shared her time between Alistair Stewart's lab and my lab here at WEHI. And so at the same time, she also managed to have 2 beautiful little girls and keep her clinical work. And so, with hard work, perseverance, determination, she really managed to be where she's at today with 2 first author publications and 2 more publications in preparation. But during all this time, she really had one constant supervisor, Dan Steinfort, at the Royal Melbourne Hospital. And I really must acknowledge Dan's work and role as a supervisor and a mentor for Tracy. We were collaborating with Dan, and he suggested that Tracy join my lab when she moved back to Melbourne. And I really must thank him for that, because it's been fantastic to have you in the lab, Tracy. I've enjoyed working with you, and I think everyone in the lab has really benefited from your time and your presence and your input. And I really look forward for more years of continuing work with you, Lou, Dan. So up to you, Tracy.
Thanks so much, Emelle. Can you all hear me? Well, as a respiratory physician, I've always had a particular interest in lung cancer, and undertaking this work has given me the opportunity to extend my clinical experience in endobronchial ultrasound, or EBUS, into preclinical applications into lung cancer. As Emelle has already described, I'll begin today's talk with the introduction into lung cancer and into EBUS, and then I'll be discussing 3 discrete but interrelated vignettes that illustrate novel applications of EBUS into preclinical research in advanced lung cancer. Part 1 will describe the molecular yield of a single EBUS pass, and then Part 2 will describe the use of EBUS samples in exploring the genomic evolution of advanced lung cancer. And then the last part will talk about the use of EBUS samples in developing preclinical models of advanced lung cancer. So back in 1912, Isaac Adler described lung cancer as being among the rarest forms of disease, where his collection of 374 cases of lung cancer represented the known global incidence. But over a century later, lung cancer is now the most frequent and most deadly cancer worldwide, and it accounts for 13% of cancer diagnoses and 19% of cancer deaths. And a major reason for this is that the majority of patients present with inoperable advanced disease at diagnosis, with a 5-year relative survival falls quite dramatically in the presence of metastases. And disease stage is not only the most important factor in determining prognosis, it also dictates the most appropriate treatment strategy. And this figure on the left depicts the thoracic lymph node stations, which is designated N1 for the hilar lymph nodes that are on the same side as the primary tumour, N2 for the mediastinal lymph nodes that are on the same side as the primary tumour, and N3 for the lymph nodes that are on the opposite side to the primary tumour. And in particular, it is the presence of the N2 metastases that is arguably the most defining aspect when you stage, because it represents a real point of divergence between treatment strategies, where curative resection is no longer possible and combination chemoradiotherapy is the recommended management. And this really highlights the importance of preclinical research into this advanced disease group to explore the drivers behind metastasis, developing accurate preclinical models, and to ultimately have a goal of identification and refinement of therapeutic strategies. However, a major challenge in the preclinical research in this group has been the acquisition of appropriate tissue samples. Traditionally, preclinical lung cancer research has relied quite heavily on surgical resection specimens from patients with early-stage non-small cell lung cancer. But because over 70% of lung cancer patients present with inoperable, advanced stage non-small cell lung cancer and small cell lung cancer, this does not represent the reality for the majority of patients. And furthermore, lung cancer is a heterogeneous disease with histological and genetic complexities, which I will define further. And therefore, tissue certainly is the issue. In terms of histological heterogeneity, 85% of lung cancer is classified as non-small cell lung cancer and 15% as small cell lung cancer. Non-small cell lung cancer is further subclassified as adenocarcinoma, squamous cell carcinoma, and large cell carcinoma. And each of these is further subclassified according to the degree of invasiveness and the predominance of specific patterns of growth. And a single tumour can comprise components of several histological subtypes. Genetic variation may also be observed. It can be seen between individuals with the same tumour type, termed inter-tumour heterogeneity, within a single tumour, giving rise to subclonal diversity, termed intra-tumour heterogeneity, and it can also occur within a subpopulation of tumour cells. There can be intercellular, genetic and non-genetic heterogeneity. The history of diagnostic tests and treatment strategies for lung cancer dates back to 1933 when the first surgical resection was performed. And over the next 70 years, progress was very slow, with surgical mediastinoscopy and positron emission tomography, or PET scanning, for staging the mediastinum being introduced in 1959 and 1974, and developments in chemotherapy and radiation therapy spanning the 1970s to the 1990s. And in the context of the challenges I have discussed, it has only really been over the last decade or so that new techniques for minimally invasive tissue acquisition, such as EBUS, have arrived in 2003, and molecular analysis with next-generation sequencing in 2005, with subsequent development of novel therapeutic agents such as tyrosine kinase inhibitors and PD-1 antibodies that target specific molecular and immunogenic pathways in lung cancer pathogenesis. In particular, EBUS has gained recognition in the last 10 years in the investigation and management of lung cancer. It is a minimally invasive bronchoscopic technique that is used to perform transbronchial needle aspiration, or TBNA, for sampling thoracic lymph nodes and centrally located lesions. It consists of a flexible bronchoscope with an integrated ultrasound probe at its tip that is inserted through the mouth and into the tracheobronchial tree. And ultrasound images are then obtained by posing the probe against the airway wall that is adjacent to the lesion of interest, and TBNA is then performed by inserting a needle through the working channel of the bronchoscope, puncturing the airway wall in the target lesion, and aspirating cells under real-time ultrasound guidance. The first few drops of the specimen are smeared for a rapid on-site cytological examination, and these are examples of an adenocarcinoma, a squamous cell carcinoma, a small cell carcinoma, and normal lymphocytes from a lymph node. And the remainder of the specimen is expelled as a cytological core of cells, which then can be processed for further immunohistochemistry and molecular analysis. And in comparison to the former gold standard for mediastinal lymph node staging, which is surgical mediastinoscopy, EBUS can sample more lymph node stations, as seen in the yellow and the pink circles, and has a superior sensitivity and specificity for the detection of lymph node metastases. Furthermore, it has a more favourable safety and cost profile, and importantly, it is repeatable in cases of disease recurrence, which is unfortunately all too common. So consequently, it has now been recommended by international clinical practice guidelines as the first-line test in the diagnosis and staging of lung cancer. So its role in clinical diagnosis and staging is very clear. But with the challenge of tissue acquisition in advanced lung cancer, we were interested into novel applications of EBUS into preclinical research specifically with regards to genomic evolution and into the development of preclinical models. So we begin by looking at the molecular yield from a single EBUS pass, where we define a pass as a distinct entry and exit of a needle into a target lesion. As I've already mentioned, recent years have seen a surge of interest in the genomics of lung cancer, with improved understanding of the pathways involved in tumour initiation and progression. And in adenocarcinoma, the commonest lung cancer subtype, the best defined include KRAS, EGFR and ALK mutations, with the development of agents that target these pathways. And this has led to a paradigm shift from being a purely histology-based understanding of the disease to subtyping now based on molecular status. But with this paradigm change and minimally invasive techniques becoming more frequently used in patients with metastatic and recurrent lung cancer who benefit most from targeted molecular agents, sampling by EBUS has gained an additional dimension where it is increasingly important for its small volume samples to yield sufficient high-quality material for multi-directional and multigenic analysis. So the challenge, therefore, has been how do we do more with less? The optimal number of passes per lymph node during an EBUS procedure to obtain a histological diagnosis has been reported, or a maximal diagnostic yield has been achieved after 3 passes. For molecular analysis, EBUS samples have been shown to be adequate, particularly for targeted sequencing of EGFR and KRAS mutations. However, unlike histological subtyping, the number of passes needed for mutational subtyping remains unclear, with published studies reporting anywhere from 1 to 10 passes. And this is of interest because molecular testing is often performed after histological subtyping, with immunohistochemistry panels has been done, and this may exhaust much of a specimen and necessitate a repeat procedure with the potential for more procedural complications and diagnostic delay. And recent CHEST guidelines published in March last year recommend that additional samples beyond those needed to establish a diagnosis be obtained from molecular analysis. But the number of additional samples is unclear. And while insufficient samples may lead to false negative results, excessive sampling has implications of increased procedural complications and increased procedure time. So we were curious as to the yield of a single EBUS pass, and we sought to explore this by 2 specific aims: to determine the DNA quantity and quality, and also the accuracy of detecting clinically relevant mutations in a single pass. So in a prospective study, paired samples were obtained from patients undergoing EBUS for the diagnosis of lung cancer. The reference sample was the routine diagnostic specimen consisting of 3 passes and processed as a formalin-fixed paraffin-embedded block, and the study sample was a single pass processed as fresh tissue. DNA was extracted from both samples and subjected to quantitative and qualitative analysis, and sequencing for mutations in EGFR, KRAS, NRAS, BRAF, and PIK3CA was performed in adenocarcinoma cases. 40 patients were recruited over 16 months, with a median age of 70 years and 2/3 being male, and adenocarcinoma cases made up 60% of these. And of these, 92% of them had advanced stage 3 or stage 4 disease, and 92% had a current or former smoking history. Sampling occurred predominantly from right-sided mediastinal lymph nodes, with representation from stations at all levels. And overall, DNA extraction yielded a mean of 3.98 micrograms, which is well above the minimum required quantity for targeted sequencing of 10 nanograms. DNA quality was measured by the DNA integrity number, and this is a numerical value which ranges from 1 to 10, 1 being highly fragmented and 10 being highly intact DNA. And the gel electrophoresis visually depicts a sample with highly intact DNA with a DIN of 9.7 compared to one with highly fragmented DNA. The mean DIN in our cases was 8.9, where over 7 is acceptable for targeted sequencing. And similarly, in the adenocarcinoma cases, there was a similar high quantity and quality of DNA. This table demonstrates mutations detected in EGFR, KRAS, BRAF, and PIK3CA in the reference and the single-pass study samples from adenocarcinoma cases. And sequencing was successful in all 24 single-pass samples. In 23 cases, there was 100% agreement between the reference and the single-pass samples for EGFR, KRAS, BRAF, and PIK3CA mutations. However, molecular analysis could not be performed in one reference sample due to insufficient tumour cells after histological subtyping had been performed. And interestingly, in this case, our single-pass study sample successfully detected a KRAS G12V mutation, which confers reduced sensitivity to EGFR antibodies. Importantly, these mutations were all detected in the context of a widely variable tumour cellularity, which is shown here, where tumour cell content ranged from 1 to 70%, and these are representative images from adenocarcinomas, the first showing a sample with 1% tumour cells, and this is the sample which was inadequate in the reference sample for molecular testing, but where the single-pass sample detected a KRAS G12V mutation. Image B and C show samples with 10% and 70% tumour cells, both carrying the KRAS G12C mutation, which was detected on both the reference and the single-pass sample. And this finding suggests that sequencing of single-pass samples is highly accurate, even in cases of low tumour cell percentage, which is particularly important in these small volume diagnostic specimens. So in summary, the number of needle passes per sampling site has implications on procedural efficiency balanced by the desire to achieve maximal diagnostic yield. And we have shown that a single EBUS pass dedicated to molecular analysis returns high-quality DNA, which is adequate for targeted genotyping of lung cancers, including samples with limited tumour cell content. And these findings have preclinical implications for utilising EBUS for more extensive molecular analysis and for the development of preclinical models. Clinical implications for performance of EBUS in cases of suspected mutation-positive lung cancer is important, where the prevailing diagnostic challenge is to do more with less. So this brings me to part 2, where we describe the use of these EBUS samples in exploring the genomic evolution of lung cancer. Previous work characterising the lung cancer genome has demonstrated countless point mutations in orange, copy number variants in green, and fusion genes in brown, with both mutually exclusive and overlapping variants among the histological subtypes. However, except for targetable mutations in EGFR, ALK, and BRAF, these efforts have not translated into major changes in clinical practice. And a major reason for this is that our understanding of the extent of genomic heterogeneity of lung cancer and the molecular events associated with metastasis are limited by a number of factors, which include the reliance on surgical resection specimens of early-stage cancers, which are not representative of advanced disease, the use of targeted gene or whole exome sequencing rather than whole genome sequencing, and the concepts of tumour heterogeneity and clonal evolution, which I will describe further. And to add to this complexity, there are characteristic mutational signatures that are associated with particular processes of DNA damage. And in lung cancer, there are 3 of particular interest. There is the age-related signature, which is characterised by C to T mutations at CPG sites, the smoking-related signature, which is characterised by C to A mutations, such as those seen in tumour suppressor P53, and the apobec cytidine deaminase signature, which is involved in RNA editing and chronic inflammation, which is characterised by C to T and C to G mutations at TPC sites. So what are the processes that give rise to these genetic profiles and mutational signatures? Well, it is thought that genomic instability through multiple pathways that disrupt the mechanisms that maintain genomic integrity result in an increased mutation rate. And this causes genetic tumour heterogeneity, which can then shape the evolution of the cancer genome, along with selection pressures by the tumour microenvironment and exposures to treatments, and this drives phenotypic variation. And such heterogeneity in advanced disease limits the efficacy of targeted treatments and poses a significant challenge to personalised cancer medicine. And tumour heterogeneity can be defined in a number of ways. There is inter-tumour heterogeneity, which is present when 2 patients with the same tumour subtype exhibit different clinical behaviours, and this might be related to host factors such as germline variants or tumour microenvironment. together with unique somatic mutations that occur within the individual patient. Intra-tumour heterogeneity describes the existence of distinct cellular populations within a primary tumour with specific genetic, epigenetic and phenotypic features that can intermingle or be spatially discrete. And this subclonal architecture can fluctuate over time in response to the tumour microenvironment or treatment exposure. Furthermore, metastatic lesions at different secondary sites can arise with different cellular populations from the primary tumour and result in heterogeneity among metastases, known as intermetastatic heterogeneity. And lastly, as metastatic lesions can acquire new mutations themselves, heterogeneity within a metastatic deposit can also exist, known as intrametastatic heterogeneity. And this can be associated with acquired mechanisms of drug resistance within the same patient. In lung cancer, intertumour heterogeneity is exemplified in adenocarcinoma, which has been shown to have widely varying genetic profiles across patients. Intratumour heterogeneity in lung cancer is illustrated here with the oval represents a resected tumour, and whole exome sequencing has been performed on several spatially distinct regions. Mutated cancer genes are to the right of the histology images, and in region R14, it has a private mutation in the ATRX gene. The numbers of mutations which are common to all regions, those are truncal mutations, or those private to each region, which are branch or private mutations, are in these windows. And you can see that there is variation in the branch and private mutation numbers between the different regions. And this leads me to the concept of clonal evolution, where the analysis of multiple samples from the same tumour can reveal spatial composition and evolutionary trajectory of subclones. Linear evolution occurs when there is sequential accumulation of mutations over time, and where a new subclone can outcompete all other subclones. Branched evolution can occur when distinct subclones evolve in parallel and result in extensive heterogeneity. Subclones can be temporally and spatially separated within the same tumour, so that biopsies from adjacent sites can be quite genetically diverse. And the clonal and subclonal composition of each tumour can be used to construct phylogenetic trees as a pictorial representation of how a tumour is inferred to have evolved. The truncal events reflect clonal mutations that are shared by all tumour cells that occur early in tumorigenesis, denoted by the blue trunk. The branch events reflect the subclonal mutations present in only a subset of tumour cells that occur later in tumorigenesis, denoted by the yellow and the red branches. The length of the trunk and the branches denotes the number of mutations that occurs in that lineage. And this is an example of a phylogenetic analysis of a surgically resected non-small cell lung cancer that has occurred in two separate regions of the lung, R1 and R3. Whole genome sequencing showed that the majority of structural variants were fully clonal and shared between the two tumour regions, suggesting that the majority occurred before tumour metastasis. In addition, there was evidence of a smoking signature, denoted by the grey box, occurring early in tumorigenesis, followed by an increase in the Apobec-related signature, denoted by the blue boxes over time. And this was more pronounced in the R3 tumour compared to the R1 tumour. So with the evidence from part one of this talk that a single EBUS pass yields sufficient high-quality material for next-generation sequencing, we sought to use EBUS samples to explore genomic heterogeneity and the evolution in advanced lung cancer with three specific aims. We looked to recruit patients undergoing EBUS to generate matched samples of primary and metastatic lung cancer, and then to use high-depth whole genome sequencing and novel bioinformatic pipelines to map the genomic evolution of metastatic lung cancer. And lastly, we looked at the clinical responses of each patient with genomic heterogeneity in the primary and metastatic sites. In terms of our sampling strategy, in patients who were treatment naive with suspected metastatic lung cancer, where the primary tumour and at least one metastatic site was accessible to EBUS, we performed multi-site sampling. One part of the sample was used to generate a cytology smear to determine tumour cellularity. One sample was snap frozen for RNA analysis and one for RNA analysis. And blood was also collected for germline sequencing. In terms of our sequencing strategy, while the majority of comparative studies of primary tumours and metastases have used targeted sequencing or whole exome sequencing, we chose to use whole genome sequencing based on the concepts that previous approaches have been heavily skewed towards identifying known early clonal drivers, which, unlike the late subclonal drivers, are almost always shared between the primary and its metastasis. Furthermore, previous approaches are limited to single types of somatic alterations, usually single nucleotide variants and insertion deletions. And in comparison to whole exome sequencing, whole genome sequencing provides highly uniform, unbiased coverage of the entire genome, and it improves the resolution of clonal analysis by mapping copy number variants and structural variants. And furthermore, at a higher depth of 150 times compared to 160 times, there is a higher sensitivity for detecting low-frequency variants at a base pair resolution and greater confidence in smaller differences in variant allele frequencies. And to date, we have successfully recruited 18 cases, 13 of which have preliminary bioinformatic results. These plots illustrate all 13 cases and the presence of deletion or loss of function in the blue squares, and amplification or gain of function in the orange and yellow squares, across a number of known driver mutations. And the first thing to highlight is that among the adenocarcinoma cases, our cohort is quite representative of the frequencies of particular mutations seen in the Cancer Genome Atlas. For example, inactivating mutations in TP53 in 60% of cases, gain of function mutations in KRAS in 30%, and gain of function in EGFR in 10% of cases. However, in the DNA repair genes, we found a higher incidence of mutations compared with other papers on adenocarcinoma, and this may be reflective of different genomic instability processes occurring in advanced lung cancer compared with early-stage lung cancer. And lastly, it was not unexpected to find several mutations in chromatin remodeler genes, and this highlights the notion that chromosomal instability plays a poorly understood role in most lung cancers. Out of the 13 cases, there were 5 where whole genome sequencing was informative in a single region. This is seen with the key where tumour regions in black indicate where 150 times whole genome sequencing was completed, in grey where 60 times was completed, and in white where, although an EBUS sample was taken, tumour cellularity was less than 10%, which is below the protocol threshold for high-depth whole genome sequencing. The pie charts illustrate the mutational patterns in that region. And to draw some examples, this is a never smoker with a stage 4 adenocarcinoma who had a largely unknown pattern of mutagenesis but yielded an L858R mutation in EGFR, which confers tyrosine kinase sensitivity. And in contrast, another never smoker, also with stage 4 adenocarcinoma, had a predominantly APOBEC-mediated pattern of mutagenesis, so this is a good demonstration of interpatient heterogeneity. But perhaps of more interest is the 8 cases where whole genome sequencing was informative in multiple regions, denoted by the black and grey circles. In 3 cases, we actually sampled 2 different regions of the primary tumour to assess the incidence of sampling bias. And in 2 cases, we were able to obtain material from an intrapulmonary and a pleural metastasis. And I'll now describe 3 particular cases for which we have the most complete data. So in case 1, this was a 78-year-old woman, a former smoker with stage 3A adenocarcinoma, who had a primary tumour in her left lower lung and a lymph node metastasis to an N2 level lymph node station. Her PET scan showed avidity in the primary tumour and in the lymph node metastasis indicative of metabolic activity, and EBUS cytology confirmed an adenocarcinoma with tiny glands indicated by the arrow. And although this patient was a former smoker, the mutational signature in both the primary tumour and the lymph node metastasis largely reflected an unknown mutagen, as well as activation of the APOBEC family of DNA-RNA editing enzymes. And from the phylogenetic tree, you can see that in the primary tumour, there were over 21,000 SNV endols and 352 structural variants. There were founder mutations that were common to lung cancer, including P53 and CDK N2A. There were a couple of mutations which were of interest, which I'll describe here. There was a loss of function in ATP7B, and this is a copper exporting gene which is usually seen in a condition called Wilson's disease. And tumours with this mutation cannot export platinum, rendering them sensitive to chemotherapy agents which are platinum-based. There was a loss of function in ERP2, which is involved in MHC class 1 molecules, and tumours with this mutation show resistance to PD-L1 therapies. There was a gain of function in ERBB2, which confers sensitivity to tyrosine kinase inhibitors, and a loss of function in POLQ, which is involved in proofreading DNA polymerase, and tumours with this mutation show sensitivity to cisplatin. And lastly, a loss of function in TSC1, which is not usually associated with lung cancer, but which can confer sensitivity to mTOR inhibitors. In the lymph node, there were an additional 9,635 SNV indels and 171 structural variants, which were private to the lymph node metastasis. And this is consistent with the idea that components of nodal disease may be established both before and after metastatic divergence. Interestingly, the lymph node metastasis gained an amplification of TERT, and this is a catalytic subunit of the telomerase complex, which is consistent with the idea that metastatic subclones can develop a series of de novo driver events under selection pressure, rather than arising from a rare subclone in the primary tumour. And in terms of clinical outcome, progression-free survival for this patient from the time of diagnosis by EBUS in March 2013 to recurrence in the contralateral mediastinal lymph nodes in November 2013 was 8 months. Several mutations present in the primary tumour had relevance for cisplatin sensitivity and resistance, and this case exemplifies the challenge of treatment choice when you are faced with biomarkers of opposing activity. This patient went on to receive multiple lines of salvage therapy with an eventual overall survival of just over 2 years. Case 2 was of a 68-year-old gentleman, a former smoker with stage 3B squamous cell carcinoma, who had a primary tumour in the left lung and also lymph node metastases at N1, N2 and N3 levels. The PET scan showed avidity in all of these areas, and EBUS cytology confirmed squamous cell carcinoma with dense keratinized cytoplasm indicated by the arrow. And in this case, it's important to point out that we were not able to obtain informative data from the primary tumour itself, as indicated by the white circle. However, 150 times whole genome sequencing was successfully performed on all 3 lymph node metastases at N1, N2 and N3 levels. And consistent with the former smoking history, the smoking mutation signature was highly evident in this patient, but there was also a significant proportion attributed to an unknown mutagen. In the primary tumour, we inferred that candidate driver events common to the lymph node metastases were present in the primary tumour, and this included mutations in SMARCA4, which is known to occur in squamous cell carcinomas, and RB1, which is characteristic of small cell carcinomas. So this combination of features is quite interesting. In the lymph node metastases, consistent with the clinical history, single nucleotide variants in 11L and 4L metastases showed strong smoking signatures and a large number of chromosomal rearrangements. However, the analysis of the lymph node metastases revealed distinct evolutionary trajectories. And you can see that in the 11L lymph node, there is amplification of PIK3CA, which is more frequently seen in squamous cell carcinomas, whereas in the 4L lymph node, there is amplification of events in ASCL1, which is more in keeping with a small cell lung cancer phenotype. And this kind of data suggests divergence in tumour phenotype can be potentially identified through genomics, with significant implications for therapy and prognosis. And in terms of this patient's clinical outcome, progression-free survival from the time of diagnosis in July 2013 to progression in January 2014 was 6 months, and overall survival was only 7 months, perhaps reflective of the presence of small cell lung cancer molecular events conferring the poorer diagnosis. And our last case was of a 69-year-old woman, a former smoker with stage 4 adenocarcinoma. In this case, we took samples from the inner and the outer aspects of the primary tumour. also from a lymph node metastasis, and also from an intrapulmonary metastasis. The PET scan shows avidity in all these areas, and EBUS cytology confirmed an adenocarcinoma with characteristic glandular structures and positivity for the adenocarcinoma marker of TTF1. And in the primary tumour, analysis of the inner and the outer aspects were very similar in terms of the single nucleotide variants and the indels, the structural variants and the copy number variants. In the lymph node and the intrapulmonary metastasis, there were multiple private mutations from the primary tumour. And this is better illustrated in a high resolution by looking at the comparative Circos plots of the 4 regions sampled in this case, where the 2 aspects of the primary tumour are at the top of the screen, and then at the bottom of the screen is the lymph node metastasis and the intrapulmonary metastasis. And you can see, looking at the 2 Circos plots from the primary tumour, they look quite similar. There are multiple focal rearrangements in the same region of chromosome 8 that are accompanied by multiple copy number variants and highly focal hypermutation in chromosome 9 and 19. And the combined pattern of focal rearrangements and copy number change is consistent with a process called chromothripsis, and the focal hypermutation is consistent with a process known as catalgesis. And both of these processes are consistent with a single catastrophic event that can occur early in the evolution of the tumour. In the metastases, there is a pattern of single nucleotide variation and structural variants that is quite distinct from the primary tumour. Firstly, most of the chromothripsis and catalgesis events in chromosome 8 and focal hypermutation in chromosome 9 and 19 is no longer present. But secondly, there are private events that include a distinct region of rearrangement in chromosome 1 and the acquisition of a large number of single nucleotide variants across the entire genome. So in terms of the clinical outcome for this patient, progression-free survival from time of diagnosis in April 2016 to progression in October 2016 was again 6 months, and overall survival was 9 months. So in summary, while there have been a handful of studies that examine matched pairs of primary tumour and lymph node metastases, these have largely consisted of early stage disease which have undergone surgical resection, and sequencing has consisted of a mixture of targeted sequencing and whole exome sequencing and only occasional whole genome sequencing. Our work has been quite novel in that it has employed EBUS to sample multiple sites in advanced disease within an individual patient, and we've used high-depth whole genome sequencing in all samples to provide the most accurate picture of oncogenesis possible. However, due to the multiple sources of heterogeneity in the host and in the tumour, it continues to be quite challenging to make broad generalisations, and so our conclusions are largely made on a case-by-case basis. Our case vignettes illustrate the heterogeneous genomic landscape of advanced lung cancer, where metastatic subclones can arise both early and late in the life of a primary tumour. And furthermore, the heterogeneity between primary tumours and their metastases can potentially impact upon treatment response, disease progression, and overall survival. And this is exemplified by case 1, where mutations implicated in both platinum sensitivity and resistance were detected, and this makes treatment choices very challenging. So an improved understanding of the phylogenetic relationship between the primary and metastatic tumours and the significance of subclonal mutations in particular and their evolution through time and space can allow clinicians to make better informed clinical choices. And this knowledge has translational implications. For example, clonal heterogeneity itself may be used as a biomarker for clinical outcome. Selection pressures like chemotherapy can select for subclones that have intrinsic or acquired resistance and allow these subclones to dominate the tumour mass and drive disease progression. And this challenges the current approaches to biomarker discovery that focus on genomic alterations in the dominant clones mainly. Lastly, there are potential strategies to overcome challenges posed by tumour heterogeneity, and some which have been put forward include targeting truncal events to limit the development of tumour heterogeneity in the first place, and lastly, adaptive therapy, whereby cancer treatment is continuously adapted in order to maintain a fixed population of drug-sensitive cells and allow overall tumour burden to remain stable. And this brings me to the last part of today's talk, which discusses the use of EBA samples in developing preclinical models of advanced lung cancer as potential platforms for testing such novel therapies. So by way of background, for decades, the mainstay of preclinical cancer research has been the use of cancer cell lines, which are cultured in a serum-based growth media and then reconstituted in vivo in immunodeficient mice. However, in these models, cells are typically derived from aggressive tumours and therefore select for a subset of patients with an inherently poor outcome. And the process of culturing cells in vitro introduces genetic and functional changes that are distinct from the original tumour and which are not resolved when the cell line is returned to growth in vivo. And this raises serious concerns regarding the accuracy of such models in predicting drug efficacy in humans. There has been an increasing focus on the development of primary xenografts, which involves a patient-derived tumour being directly implanted into immunodeficient mice. And these have been shown to retain the principal characteristics of the original tumour in terms of histology, gene expression profiles, metastatic capabilities, and response to treatments. And these changes are retained even after serial passage across several generations of mice. And as such, these models possess enormous translational potential. They have the ability to expand tumour cells and constitutes an important repository of biological material from small volume samples where surgical resection is not possible. They can also be applied as an in vivo screening tool to test novel anti-cancer treatments. They can be used to evaluate key markers of response and resistance to conventional and investigational agents. And lastly, they can facilitate individualisation of treatment regimens by preclinically assessing the sensitivity of tumours to anti-cancer drugs in vivo and in clinical trials with humans as so-called mouse avatars. But to date, most of the studies that involve PDX of lung cancer have used surgically resected specimens. And as I have repeatedly mentioned, the majority of lung cancers are inoperable. And this is particularly the case in small cell lung cancer, which grows rapidly and metastasizes early, often to mediastinal lymph nodes. And it has the highest lethality of all lung cancers with a 5-year survival of 5%. And while platinum chemotherapy and radiation therapy are the mainstays of treatment, this has not materially changed in the last 3 decades. And disease recurrence is near universal, often with the development of treatment resistance. Therefore, in this particular patient group, there is a significant need for accurate preclinical models to improve the understanding of the biology of the disease, with the hope of developing new therapeutic strategies. And EBUS is particularly well suited as a sampling technique in this group, and we were interested as to whether PDX could be developed from EBUS samples of small cell lung cancer, which had not been performed in the past at the time of this study. We had 2 specific aims. The first was to recruit patients with advanced small cell lung cancer undergoing EBUS to prospectively generate PDX, and then to determine the histological, immunohistochemical, and molecular fidelity of the PDX to the primary tumour. From the EBUS sample, 1 portion was taken for histological, immunohistochemical, and molecular analysis. 1 portion was resuspended in matrigel and injected into the flank of a non-skid gamma mouse and monitored for tumour growth for up to 6 months. 1 part was taken again for histopathological and immunohistochemical analysis and targeted next-generation resequencing for comparing with the primary tumour. And furthermore, 1 part was disaggregated for reimplantation in nude mice and serially passaged. We recruited 12 patients with a mean age of 64, all with a positive smoking history and advanced stage in 92% of cases. EBUS samples yielded large numbers of viable tumour cells, sufficient to inject up to 1.5 million cells per flank. The engraftment rate was 83%, with a mean latency of 104 days, and this is in contrast to reported engraftment rates of 20 to 40%, with latency periods up to 300 days. And of our 10 successfully engrafted small cell lung cancers, histological concordance was seen in 90%, where 9 retained the typical small cell lung cancer phenotype, but 1 interestingly resembled a large cell neuroendocrine cancer, and this might have been representative of an outgrowth of a subclone in a heterogeneous primary tumour. the neuroendocrine immunohistochemical staining with synaptophysin and CD56 was concordant in 100%. It's important to point out that 3 of the primary tumours unfortunately did not have staining for comparison. And in the 10 cases with successful engraftment, comparative targeted resequencing was performed between the primary tumour and the PDX. And this plot shows the primary tumours with their corresponding PDX. It shows the genes sequenced down the side. The red boxes indicate if there has been a frameshift mutation, the orange boxes if there has been a nonsense mutation, a brown box if there has been a missense mutation, and a blue box if a homozygous deletion was detected. In our cases, the mean percentage of the coding region variants shared by the primary tumour and the derivative PDX was 81%. And this indicates that to a large extent, the PDX recapitulates the genotype of the primary sample, particularly with regard to the well-described driver mutations. There was, however, discordance for several mutations that existed in 7 of the paired samples. The blue arrows indicate the mutations which were present in only the primary tumour, and the green arrows where the mutations were present in the PDX. And I'll just draw your attention to PDX 105, where interestingly an RB mutation characteristic of small cell lung cancer was detected that was not present in the corresponding primary tumour, but only in the PDX. And this again suggests that the process of xenografting may select for genetically distinct subclones that are derived from a highly heterogeneous primary sample. So in conclusion, EBUS samples generate large numbers of viable tumour cells that are suitable for efficient xenografting, and also high-quality DNA that is suitable for next-generation sequencing. Small cell lung cancer PDX lines that are derived from EBUS retain characteristic features of the primary tumour, and as small cell lung cancer is largely inoperable, the provision of engraftable samples by EBUS has the potential to dramatically expand the potential for preclinical modelling in this highly lethal disease. So in final summary, with the advent of EBUS, we are now able to obtain samples from patients with inoperable advanced lung cancer, and we are able to sample their primary tumour and metastatic sites. It requires minimal additional sampling during a diagnostic procedure to obtain material for genetic research purposes. And in this way, a new avenue has opened up by which we can explore the genomic landscape of advanced lung cancer with the use of advanced next-generation sequencing and bioinformatic approaches. Furthermore, EBUS samples can successfully generate preclinical models, such as PDX, and offer an unparalleled opportunity to test therapeutics, which may be developed as a result of genomic evolution and biomarker studies. However, the work presented here today is really only the tip of the iceberg in terms of what can be discovered, and we look forward to ongoing work with the valuable samples obtained by the technique of EBUS. So that brings me to my acknowledgements. There are many people to thank, as this was very much a team effort. So firstly, I'd like to thank my primary supervisor and clinical supervisor, Dan Steinfort from Royal Melbourne, and the team from Royal Melbourne for starting the journey with me 5 years ago and navigating me through very uncharted waters. I'd like to thank Marie-Liesse for welcoming me to WEHI and taking on a fairly unknown entity by welcoming me to her lab, and also for coming back from the States to be here today. I'd like to thank the past and present members of the Asselin Labat and the Sutherland Laboratories for their ongoing help, and in particular I'd like to particularly thank Claire Whedon and Casey Khan for answering my very naive doctor questions throughout all of this. I'd like to thank the rest of the stem cells and cancer division and the supporting facilities throughout WEHI for all their help, not just with these projects, but with others which unfortunately could not be presented today. I'd like to thank my co-supervisors, Professor Alastair Stewart from the University of Melbourne and his team from the Department of Pharmacology and Therapeutics for their help with some of our therapeutic ultrasound work, which is ongoing. And Professor Neil Watkins, who unfortunately could not be here today, but with his team at the Garvan Institute, and also with the clinical team at St Vincent's Hospital in Sydney, who help us a lot with the sampling for bioinformatic projects. I'd next like to thank the Victorian Cancer Biobank team, particularly Leanne and Catherine, without whom we would never be able to obtain the samples that we do to perform this work. And I'd like to thank our collaborators with Peter McCallum and also with the Hudson Institute as well. And lastly, I'd like to particularly thank the patients and their families who are so generous in their time and in their samples at what is an extraordinarily devastating time in their lives. And lastly, but not least, I'd like to thank my family, my husband Paul and 2.5 children, for what has been a very long journey. And all I can say is that we're nearly at the end of it. Thank you.
Thank you, Tracy.
Thanks,
questions for Tracy? Yes, right at the back.
Just with the group cases that you found that there was a mutational signature, were there commonalities between the 3 different cases with that underinfection? Like, is it possible that there's a unique lung cancer mutational signature that we know about yet?
It's a very tantalising thought that, similar to other cancers, and colorectal cancer comes to mind, that there would be a path of sequential mutations that we can identify. But unfortunately, lung cancer, as it's been illustrated by multiple papers, is so highly heterogeneous that it is very difficult to draw a generalised view which is going to be common to all patients. And so what we hope, and the eventual aim and the ongoing work with this project, is to almost build up a bank of different types of histological subtypes where we can draw from information drawn from each of these just to inform us. But unfortunately, I don't think we ever will be able to find a common mutational signature. Lou.
Tracy, this is fabulous work, and on the point that was raised about the unknown drivers, this is incredibly important because although someone smoked, it doesn't necessarily mean that their lung cancer has been caused by smoking, because over 50% of the population used to smoke. But at the moment, there are enormous biases against people who smoked, and the outcomes are highly heterogeneous. And one of the questions I was going to ask is that there's new evidence that from large epidemiological studies that atmospheric pollution, in particular diesel pollution, is a very strong causation factor for lung cancer. And I'm just wondering if you know whether there are any molecular signatures of that, or whether you've got a way of exploring that.
It is really interesting you bring this up, and for those who don't know, certainly where literally over the last decade or so, the epidemiology of lung cancer has been changing from being a predominantly smoker-related disease to now affecting patients of increasingly younger age, females who have never smoked. And in particular with occupational exposure, to my knowledge, it has not been directly assessed. I'm sure that particularly there is a large study, the TracerX study, which has been performed looking at longitudinal sampling of patients with lung cancer. I'm sure they would be obtaining a lot of epidemiological data there about occupation. But interestingly that you mention, in this discussion with Neil Watkins last week, we are also thinking at looking at this ourselves. So it's very important.
Thanks for a lovely seminar.
Thanks, Jeff.
So I'm just curious with these single pass versus the quality of the RNA that you can extract as well, or whether that should be a subject of your next PhD. Given the increased emphasis on immunotherapy, what are the prospects for characterising the immune profile for EBUS, and what are the contamination issues with the lymph node?
Absolutely. With this particular study at the time, we were particularly interested in DNA just because it was what was clinically actionable at the time when we did the study a number of years ago. But interestingly, with the multi-site sampling study, we actually are performing in parallel RNA sequencing because the transcriptome is so important, and because immunotherapy, which I'm sure many people will know, is now sort of the newest hope for lung cancer. So there is definitely an interest in doing this, and we are actively doing this in my next PhD.
The analysis of the primary and the lymph nodes led to quite different profiles in many respects. When you put them in the PDX, they didn't change very much. So still a long latency period. Why are the PDX stable and the lymph nodes so different to the primary?
I think that it's probably a twofold answer. The first is that we, for ethical reasons, did not wait for metastases to occur in the mice. Had we waited long enough and been able to sample metastatic lesions, then possibly we might have seen private mutations occurring in lymph node or distant organ metastases. And I guess the second point is one which I think I demonstrated with comparative CIRCOS plots. We performed only targeted sequencing of known driver mutations in the PDX, and it only picks up a proportion of known driver events, whereas in the multi-site sampling study, we really wanted to get that very high resolution by doing whole genome sequencing, which both confounds and illuminates us because it picks up so many more mutations and shows up private mutations which may not be seen just on the targeted resequencing.
Just on that, I mean, the TCGA is having to go back and resequence because they've got so many errors from the
Yes.
sequence. What's your guarantee that the sequencing is halfway reasonable?
I guess there are no guarantees, and if the TCGA is reporting errors, it would be difficult for any of us to sort of, we all have to really question the validity of our own work. And this is where validation with RNA sequencing will be important, because we do want to look to see if the findings that you find in DNA changes correlate with what we see, which has been transcribed. So that, as Jeff has pointed out, is going to be a very important step.
Just along those lines, it was a very nice demo.
Thanks, Jane.
Just interested in the chromosome remodeling genes and the mutations there. So does your data from those 3 patients correlate with that seen in the TCGA? And also, are those same genes implicated in small cell lung cancer?
So in terms of comparing with TCGA data, yes, is the answer. There has been this long-standing theory that chromosomal instability bears quite a significant role in lung cancer. We're yet to really define exactly what chromosomes and what particular mechanisms of chromosomal instability are implicated. In terms of small cell lung cancer, we've to date only recruited 2 patients where we've been able to sequence multiple sites, just of the nature of how unwell these patients are at the time of their diagnosis. And we're yet to actually get preliminary data on that, but it will be a very interesting thing to see whether chromosomal instability in those particular patients is a significant finding.
Last question.
I guess another difference between the humans and the mice are their immune systems, or
You were going to say that? Yep.
And so is this a model where you can actually investigate what happens with it without the immune system metastatic lung cancer?
I mean, I think that, there are people in this room who know more about this than I do. One reason for us to serially passage the tumours in more immunocompetent mice, which are the nude mice as opposed to the Nodsky gamma mice, was to try to, I guess, recapitulate a more immunocompetent host. But at the end of the day, you're right, the microenvironment is different from what a human will be. And we know that the tumour microenvironment plays a very significant role in tumour progression. So that is something which does need to be borne in mind.
Well, I think it remains to thank Tracy for a fantastic seminar.
Just on behalf of the Department of Medicine, where you did that.
Yes.
Thank
you, Prof.
to acknowledge this presentation.
Thank you very much, Prof. Cheers.